A hairy truth about your sense of touch. Researchers showed that cells in your hair follicles release important chemical messengers in response to gentle touches to your skin. When someone brushes a hand across your skin, it's like a breeze blowing through a forest of countless small hairs. Nerves that surround your hair follicles detect that contact, and very far away in your brain, other cells fire. Some of the neurons responding to light contact might make you shiver and give you goosebumps. Some might tell you to move away. Or they might tell you to move closer. Scientists who study the sense of touch have explored which cells bear these messages, and they have made an intriguing discovery. Follicle cells triggered by hair movements release the neurotransmitters histamine and serotonin, chemical messengers linked to biological phenomena as varied as inflammation, muscle contraction, and mood changes. The observation, reported in October in the journal Science Advances, lays the groundwork for tracing how gentle touch makes us feel the way it does. Studying hair follicles is challenging, because they begin to decay soon after being removed from the body, said Claire Higgins, a bioengineering professor at Imperial College London and an author of the study. So she and her colleagues went to a hair transplant clinic. There, they were able to look at freshly harvested follicles, which they gently prodded with a very small rod to simulate touch. The scientists knew from work done by other groups that the neurons in the skin surrounding hair follicles are capable of sensing movement. When you brush your hair, you feel it because the sensory neurons are directly being stimulated, Dr. Higgins said. But they were curious whether the cells of the follicle itself, the tube from which a hair sprouts, could be contributing to some of the feelings associated with more gentle touch. Not all of the follicle cells had movement sensors, but some did. The researchers identified these and watched them carefully as the rod touched them. We found that when we stimulated our hair follicle cells, they actually released mood-regulating neurotransmitters serotonin and histamine, Dr. Higgins said. Blocking the receptors for these neurotransmitters on nearby neurons meant that they no longer fired when the hair was stroked, confirming the link between the follicle cells and the neuron's response. Just because these neurotransmitters are associated with mood in the brain does not mean that they are linked to emotion elsewhere in the body, Dr. Higgins said. They are messengers, and the nature of the message they carry depends on which cells they are stimulating. Skip advertisement. But she points to research by Francis M. Sloan, a neuroscientist at Liverpool John Moores University in England who has studied the rewarding feelings we get from touch. He and his colleagues have identified nerves in the skin that respond to gentle touch, generating that warm glow we get from human contact. Were the neurotransmitters being released by follicle cells in this study stimulating those nerves specifically? No one knows, but Dr. Higgins hopes future work will illuminate the identity of the cells the neurotransmitters target. She is curious how increasing levels of serotonin or histamine in the skin might change what happens in the brain, at the other end of the transmission. In the tiny sheath of cells containing each hair, there may be answers to questions about something as fundamental as human connection. The follicle never ceases to amaze me, she said. Mechanical stimuli, such as stroking or pressing on the skin, activate mechanoreceptors transmitting information to the sensory nervous system and brain. It is well accepted that deflection of the hair fiber that occurs with a light breeze or touch directly activates the sensory neurons surrounding the hair follicle, facilitating transmission of mechanical information. Here, we hypothesized that hair follicle outer root sheath cells act as transducers of mechanical stimuli to sensory neurons surrounding the hair follicle. Using electrochemical analysis on human hair follicle preparations in vitro, we were able to show that outer root sheath cells release ACT and the neurotransmitters serotonin and histamine in response to mechanical stimulation. Using calcium imaging combined with pharmacology in a co-culture of outer root sheath cells with sensory neurons, we found that the release of these three molecules from hair follicle cells leads to activation of sensory neurons. The hair follicle is a complex dynamic structure that is present in the skin of all mammals. Hair follicles have many functions, such as thermoregulation, sweat production, and skin protection. 1. In rodents and other mammals, vibrissa, whisker, follicles have known roles in mechanoprocessing. 2. Humans lack whisker follicles and have instead developed our sense of touch, which enables both physical, navigation and perception, and emotional, pleasure and neurodevelopment responses. Low-threshold mechanoreceptors (LTMRS) are sensory neurons present within the skin that transfer tactile information to the brain. In glabrous skin, non-hairy skin located on the palms of our hands and soles of our feet, this transfer facilitates an understanding of our external surroundings. 3. 4. Hairy skin also contains CLTMRS, a specific type of tactile neuron that is not present on glabrous sites and is involved in processing affective touch response. 5. 6. Various mechanical stimuli, such as stretch, skin movement, compression, or hair fiber deflection can activate these different classes of mechanoreceptors within the skin. 4. 7. While mechanoreceptors were originally assumed to be located on the sensory neurons, recent data have shown how Merkel cells and keratinocytes, both located in the skin epidermis, play a role in touch perception and can also be classified as mechanoreceptors. 8-10. While Merkel cells release noradrenaline and serotonin in response to tactile stimuli, keratinocytes release adenosine 5-triphosphate ATP. In turn, these molecules activate sensory neuron afferents associated with the Merkel cell complex and skin epidermis, respectively. 8, 11, 12. After activation of mechanoreceptors, sensory nerve endings within the skin are then involved in sending tactile information to the brain. In mouse skin, the physiology and morphology of these nerve endings have been described in detail, revealing that in addition to the skin epidermis, hair follicles are innervated by a heterogeneous group of sensory neurons with longitudinal and circumferential morphology. 4, 13, 14. While several groups have shown the presence of axons around human hair follicles, a clear picture of their molecular identity as well as their longitudinal and circumferential and organ morphology has not been described. 15-18. Despite the complex innervation patterns localized around the hair follicle in hairy skin, mechanotransduction capacity has not been attributed to cells within the hair follicle. We hypothesized that hair follicle epithelial cells, which we shall refer to hereon as outer root sheath, ORS, cells, act as a transducer when the hair fiber is deflected, facilitating the activation of sensory neuron afferents that wrap around the follicle. In this work, we used a combination of immunolabeling, calcium imaging, and voltammetry on in vitro and ex vivo preparations of human hair follicles to investigate this system's role in processing tactile information. Humans have morphologically and molecularly distinct mechanoreceptors innervating terminal hair follicles. In human scalp, hair follicles grow in follicular units with 3 to 4 hairs per unit. Unlike mouse skin, where there are several different types of follicles, in human skin, hair follicles are classified as either terminal or vase. To assess the architectural arrangement of circumferential and longitudinal nerve endings, 19, around terminal human hair follicles, we performed whole mount immunostaining with volumetric and three-dimensional 3D imaging in cleared human follicular units taken from occipital scalp skin, Fig. 1 and Fig. S13. First, to investigate LTMR innervation, we used an antibody against the neuronal marker neurofilament heavy (NFH) chain, which is expressed in circumferential and longitudinal large and medium diameter LTMRS20, combined with an S100 antibody, which binds to myelin-forming Schwann cells, Fig. 1b21. We found that human follicular units are innervated by NFH-LTMR subtypes with localization around and above the K15 region of the hair follicle that contains hair follicle stem cells, Fig. S1. These NFH neurons formed both circumferential Fig1C and longitudinal Fig1D endings and a proportion had S100 myelinated afferents. Consequently, these neurons can be classified as either longitudinal LTMR, or circumferential field LTMR, a newly found LTMR that is responsive to skin stroking. 22. To better assess the structure and shape of the NFH nerve endings, we used high-resolution confocal microscopy with 3D rendering and identified circumferential Fig1E and lanceolate Fig1F structures in a collar around the upper permanent region of the hair follicle. With NFH staining, it is not possible to distinguish between longitudinal LTMR and longitudinal LTMR. Therefore, to specifically assess whether a LTMR were present around human hair follicles, we used an antibody against RET receptor, which marks a LTMR, alongside an FH. We found that these antibodies labeled neurons that form longitudinal nerve endings around human hair follicles, which match the features of a LTMR found in mice. To further investigate the morphology of other tactile receptors around human hair follicles, we expanded our whole mount immunostaining analysis.